Good morning. Good morning, respected principal sir, the chief guest of the today's event, faculty members, online and offline students, and the faculty attending the 3-day FPP on AI in drug discovery and development. Myself, Antaras Panigrahi, working as assistant professor in Drug College of Pharmacy, welcome you to this forenoon session of day 2 on a 3-day hands-on workshop in AI in drug discovery and development. So, with this small introduction, I now invite Dr. P. Mallikarjun sir and hand over the forenoon session of this 3-day workshop. Welcome, sir. Welcome to this session.
Hi, good morning. Uh, okay, today's session is going to be, uh, I'm going to show some databases, okay, in which I'm going to talk about 20 to 25 minutes about what is the database, how it is going to help in the drug discovery process, okay, and what information that you should obtain from those databases. Okay, some of the tips and tricks, okay, to get the useful information, and also the common mistakes, usually what the people do. Okay, so we are starting with a gene code, followed by a PubMed, followed by a string. Okay, these three related to a biology. Then afterwards, we are going for AlphaFold, protein data bank, Campbell, and PubChem. Those four are categorized under something related to drug discovery. Okay, before you go into a drug discovery. Okay, and the last part is about when you are optimizing the compounds. Okay, chemical compounds. Okay, for to exhibit better activity, we go for Swiss ADME and Swiss similarity. Okay, so I request you all, please pay attention. Okay, and because there is a lot to cover, but I am making it bit crispy. Okay, so that tomorrow session will have a better interaction, discussion. Maybe whatever you learn, if you have any questions, it's easy to ask. Okay, then the first one is about the gene code. Because in yesterday we thought that we discussed that to start a drug discovery process, first thing is we should have a molecular target. Then the question is where should we obtain that information from the databases, and that database is the gene code. Okay, gene codes help researchers build a structured target dossier by integrating gene identity, protein function, pathways, disease links, variants, expression, and literature evidence. Okay, that means with these gene cards, actually you can get all the information, and lots of information is available, and every day it is being updated. Okay, so why this matters is target validation starts with gene level evidence. Here the key point is a target validation. Okay, validation means when we take a particular target, molecular target, we should know that yes, it is really applicable in that particular disease. Okay, that is target validation. Aliases and identifiers prevent database errors. That means a particular gene will be called with different names, okay, in different databases. So if we use only one particular name, okay, then we may miss some of the literature. So we always need to know what are the aliases and the identifiers, and we need to know that this target is implicated in what disease and what are the different variants that are there for that particular gene, which is called as the mutants. Okay. Then the expression data flags efficacy and safety questions. That means we also need to know that where the particular gene is expressed, in which organ. Suppose all of you are working on this beta adrenergic receptors, okay, and you know that they work on the cardiovascular system and also they work on the respiratory system. That's why, of course, I don't want to go into details. That's why most of the cardiovascular drugs also have the side effects on the respiratory system. Okay, and the drugs that are used for the respiratory diseases also have some side effects on the regulatory system. Okay, sorry, on the cardiovascular system. And if you look at the structures, they look very similar, but there are small changes. Okay, so this is also important. Okay, then EGFR example. Okay, if you see that EGFR, it is also called as these two names. Okay, so when you are searching for a literature, you should search for a literature for EGFR, ERBB1 and HER1 all together. Okay, that's why, of course, nowadays the people say that we can use AI. Yes, we can use AI, but AI won't tell you if you put only EGFR, it won't search for these things. Okay, that's why when we write a prompt in using a generative AI, we must know that actually what prompt should I write. Okay, then encodes a receptor tyrosine kinase. Okay, and mutations drive selected cases because different patients suffering with a lung cancer, they will have a different mutations. That means they respond differently to the different medicines. Okay, and of course, approved the EGFR inhibitors validate the target clinically. Suppose in drug discovery process, if someone started saying that I'm completely working on a new target, it's very difficult for the people to believe that that project will be successful. Okay, because in a research defense, okay, always we need to show some scientific evidence. Okay, otherwise, actually, no one will support that process. Okay? Then learning goal is using gene cards to ask whether a gene is biologically credible, druggable, and clinically relevant before starting a compound design. Here, druggable means every protein we should identify whether it contains an active domain or not. Okay, and then you need to see that whether the active domain have a space or not. And in that space, you can go into whether you can put a molecule. That is called as a druggable. All the proteins are not druggable. That's why for some diseases, there are no medicines at all because those proteins are not druggable. Okay, actually, for that, in medicinal chemistry, there is a completely different topic that is called as PROTACs, protein-protein interaction inhibitors, molecular glues. Okay, these are the new techniques that the people are using. Okay, for targeting those proteins which are not druggable. Okay, then gene cards is a target intelligence platform, not just a gene dictionary. Okay, then next one. Okay, then where gene cards fits in the drug discovery pipeline from disease question to the target dossier. So it will tell you whether in a particular disease it is required or not. Then the target identity, confirm the correct human gene and aliases. Because if you don't get this correct human gene, okay, you won't proceed. And here the key point is a human. because genes will be shared by more than one animal. Human is also an animal. Am I right? So we need to look at the humans one, and we need to understand the biology. That means we need to understand what is the function of it, and in what are the disease pathways that particular gene is involved, and what are its interactions with other genes. And a clinical link, you see that whether that particular protein is linked to a particular disease or not. Then later on, we are going to talk about string, AlphaFold, Campbell, and PubChem. Then later on, that means whatever the information that you get from here, you are going to use that information in the later that we are going to talk about these things. In that way, you can see some kind of a link between the different databases. Okay, gene cards help all these answers. What is the gene and protein? Okay, what are the IDs? What pathways it is involved, and what are the variants? Okay, but what gene card does not prove is a causality in a disease. That means when that particular gene is present in a particular disease, okay, gene card won't tell you that okay how deadly the disease is going to be. It won't tell. Okay, that is only in the clinical practice, and it won't tell you okay whether the drug how effective it is. It will tell you that particular compound may interact with it, but it won't tell you whether it is effective or not. And it also won't tell you that whether the experimental assay is going to be successful or not. It won't tell you. Okay, that is completely different one. And it is also do not tell you that safety. When you modulate that particular protein, and gene cards won't tell you that whether the compound is going to be safe or not. And also it also do not tell whether it is going to be clinically useful or not. Okay, this is one. Because why I'm telling this one is you are in a drug discovery research. You should know what it is good for and what it is not good for. Okay, and best mindset is use gene codes to organize evidence, then verify important claims in primary databases and publications. That means whatever the information that you get from these gene codes, you need to again confirm with other things. Okay, gene codes provide the map. The researcher must judge the evidence. Ultimately, you are the key person to judge whatever the information that is there in the database is correct or not. Still, it depends upon the human intelligence. Next step. Okay. Then a structured overview of gene as a potential therapeutic target. Okay, this is what we do. Okay, official gene symbol, cross database identifiers. Okay. Later on, yesterday, I think you have received some of the workshop material. In the workshop material, all the information is there. Again, I'm telling you that it's only for you to make you understand. Okay, actually, for each database, for you to fully understand, in my view, it will take minimum 100 to 150 hours to fully explore. It is not that I can deliver it in half an hour and you can master it in half an hour. Okay, but actually that database, whatever the manual that we have created, actually it is keeping in our mind that it is for early career researchers. Okay. Then chromosomal location and transcripts and protein products. Okay, that is the target identity. It will also talk about the biological context. Okay, then it is about the clinical context. So here you can look at this one. This is called as because drug discovery is expertise from the different fields. So this one is a molecular biology field. This is a pharmacology field. This is a medical field. So when these three work together, then only the drug discovery will process happens. What happens, but in general, if one person only talks about thinks about all these three, and it will be overburdened. Okay, then a useful target usually integrates identity, biology, disease relevance, and clinical evidence. Okay, and most of the time, what happens is the doctors don't speak to the researchers, and researchers don't speak to the doctors. Okay, doctors always think that my job is only to treat the patient. Okay, and researchers think that my job is only to discover something without understanding actually what is happening in a clinic. Okay, this is the beginner person. Okay, what is BGFR? I feel that most probably the students who are here, with the due respect, I expect that you all are at a beginner level. So, what is EGFR? These are the questions that you need to understand. Because the most important thing is before you do something, Don't simply search the database. You should have a particular question. Actually, what for I'm looking for, because there is a lots of information. That's why many researchers, what I know is they say that this database is good, that database is good, this tool is good, that tool is good, but they do not know what they really want. Okay. Okay. So everything we need to start with the question. Okay. Actually, what it is, where it is located, what protein it is this one, and what this is. Okay. This is very important. And drug discovery question. From this one, you will write the drug discovery questions. And these are called as the research questions in any kind of a research proposal. Okay. Is EGFR disease driving or really associated? Okay. That means in a particular disease, some proteins are there, but they do not play any role. Okay. Then whether you modulate, nothing will happen. Okay. Then is the encoded protein druggable? Okay. And is there mutation or biomarker evidence? Okay. And is the target clinically validated? And what is the intended end use? This is very important. And most of the time, except in pharma industry, okay, no one actually think when they write a research proposal, what is the use, what is the end use in the clinic, okay? And these are the end use example. Okay, this is the research aim, okay? Because in any research proposal, we have to start with a research question, then there should be a research aim. So this is the research aim. Identify orally active mutant selective EGFR inhibitors with acceptable ADMET and CNS exposure for resistant non-small cell lung cancer. Because this is very clear. Because from this one, what we want is, we want the drug to be orally active. That means it should be in a tablet form or in a capsule form or in a syrup form. Okay, that's what we want. And we want the drug to be mutant selective. Okay, because we don't want that to, some people simply say that selective. Selective with respect to what? Okay, so we need to explain mutant selective. Okay, and we also want to say, some people will say good ATMET properties. What is meant by good? There is no definition. And here they say that acceptable. Acceptable means, yes, it should not, you may not meet 100%, but you need to compromise. Drug discovery is always a compromise between the efficacy, safety, and toxicity. Okay, then, and CNS exposure. For some diseases, we don't want the CNS exposure to happen, but for some diseases, CNS exposure is required. What do you think that in EGFR, in lung cancer, is CNS active we want or we don't want? We want, because there is a metastasis. Okay, so good. Okay, this same tells you what evidence to collect. Okay, then the best strategy is guided by the candidate profile you ultimately want. That means if you are not clear and you do not know what to do with it. Okay, next slide. Okay, then best way to set theme cards. Okay, okay, start with a official symbol. Okay, official symbol means EGFR first, then check aliases such as this one, all these symbols. Then confirm organism, and please make sure that you select only human being. Okay, don't select other things. Okay, and read summaries critically. Actually, you need to read and read and read and read. Okay, and that one, please don't take the help of any kind of AI platform to read for you. Okay, because ultimately AI platform is not going to do an experimental research. Okay, until now there is no AI platform who can go to the lab and do the research. Okay, ultimately at the end you are the one who is going to do this one, so you need to understand that. Then extract structured evidence. That means you need to write a notes. You need to write a notes. Some people simply they glance through, then read, and you need to read out what are the points. Okay, then follow source link. Okay, then verify the key facts in the all these codes, Uniprot, PubMed, Campbell, PDB. That means you need to cross check them. Then write a target dossier. Okay, this convert fields into a short rationally explaining why the target matters. So this is the one, target dossier is one. When you are applying for a research grants, okay, that the people who are there evaluating your research proposal always try to prove you wrong. Okay, always try to prove you wrong. That is their job. Okay, so our job is to make sure that they agree whatever we say. So this is the one. Okay, so this is really happens in the drug discovery process, and recommended search phrase examples. Even for example, only for EGFR, actually we need to search for all these things: EGFR, ERBRB1, HER1, EGFR, NSVLC, EGFR exon 19 deletion, EGFR this mutation, this one. You cannot simply put EGFR. If you put EGFR, the people think that you will get everything. And this is the one of the greatest mistakes what the people do who heavily relies on platforms, generative AI or AI platforms. Okay, search with aliases, but regard one ambiguous official identity. Okay, it should be unambiguous. Next slide. Okay, and a practical navigation map for a target dossier. Okay, this is not the menu section, because later on you are going to, when you open the grade cards, this is what you will see on your left hand side, and actually what it tells each and everything about. Okay, basic gene and protein description, alternativeness, chromosome, protein products, biological role, where the target is expressed, clinical links, and what is its use. Because we must be very clear. Okay, whenever you are searching for it, you need to know where you have to search for, and there is a lots of information, but you need to know that what exactly you need to look for. Okay, then you need to see that where it fits into your drug discovery plan. Okay, then rapid orientation and the target rationale, cross database searching, mutation and cancer genomics, druggability, mechanism, drug accessibility, precision medicine, and clinical validation. As I told you that this precision medicine is also called as a personalized medicine. I think in another 5 years or 10 years, everyone is going to talk about this one. Okay, that's where the pharmacists are going to play a huge role. Okay, and here, do not read gene cards randomly. Okay, navigate according to the evidence you need. That means you need to have a particular question why you need, why you are looking for it. Okay. No, finish it. Okay, next one. Okay, here. Okay, and here this is dossier field, so it avoids the confusion across papers and databases, because different people are using different mutation names. Okay, then how to use it later. So whatever the information that you got, you search it in PubMed, String, PDB, or Campbell. Okay, and then you need to know about the NCBI gene. Okay, it provides unambiguous database identity. Okay, that gives uniprot number. Okay, for the, there is a specific identity number. Then you need to know the protein type, what it is, and of course EGFR kinase inhibitors, and you need to know the pathways, and move into the string and pathway analysis, expression and localization, and suggest accessibility. Okay, and understand skin and GA effects of EGFR inhibition, because most of the cancer drugs. Okay, and how many of you have seen the cancer patients who are undergoing chemotherapy? How they look like? They look like a pathetic, am I right? That's why majority of the cancer patients, what they say is, I want to live happily as long as I live. I don't want all these medicines. Because vomiting, okay, diarrhea, hair fall, okay, looking very thin, and because human always they want to see us healthy. Once you start a chemotherapy, it's a nightmare. It's a really nightmare. Until now, that is the best research gap. That is really the best research gap that anyone can do it. Okay, it's not about discovering the medicines. In my view, how to make the cancer patient happy to take the chemotherapy. That itself is the biggest research question. And at this point of time, China is the number one in the world. China is the number one in the world. The people who are undergoing cancer, okay, they will go to the China, and within 1 month they will come back with fully a perfect person, full of hair, everything will be there, energetic, okay, healthy, and all those things. That's why, in one way, the China is very well ahead of what others do. Okay. Then, of course, always you need to look at our publications. Should support the next experiment, not merely describe the gene. Okay. So this is about this one. Okay. Okay. Good. Then you have about you go through this. We'll stop here. You go through whatever the material that was given to you. Just search for it. Okay. And don't try to because at this point of time, don't try to memorize everything. Just go through it. So you will have about 10 to 15 minutes time. Okay. Then whatever the questions that you have, you write somewhere. Then later on we are going to talk about it. Okay. So yeah, she's going to show you this. Okay. Actually, this entire workshop material is prepared by one of my colleagues, one of my colleagues, and she was trained in drug discovery under my supervisor, and she is very meticulous. She is very meticulous, and until 2 years back, I am her mentor, and in these 2 years, she is my mentor. Okay, she teaches a lot to me. Arjun, this is wrong. This is wrong. This is wrong, and she became the top 2% scientist by the time she completed her PhD. Woman. All these things was prepared by this one. Actually, you can see that the way that she prepares the manual is really fantastic. Okay. 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 So here, how to use this manual? Okay. This one structure. This one. Go to this one. Go to the next one. Of course, this is what about this one. Next. Yeah. Here you can see that what are the learning objectives because you need to know what you have to learn. That what is the background. Of course, it was given some kind of a breakthrough. Of course, I already have given an introduction. Then, actually, can you go to the step-by-step procedure? Yeah, please follow these things. Huh? Okay, just go around for 15 to 20 minutes. Again, uh, we are going to start the next session, PubMed, by 10:20. Okay, okay, thank you. No, afterwards, we are going to string. Afterwards, we are going to AlphaFold protein data bank. That means each session, first I'm talking for some time, then
I, hello. I request all the participants to open the file shared in this chat box so that our speaker can clearly explain regarding the utilization of these gene cards. Thank you.
Okay, madam, just open the PDF document. Okay, here, can you go to the, just to follow whatever it is stated here, say, open your browser and go to the genecards.org. You do that. Then in the search tools, type EGFR and click search. Okay. Then on the summaries tab, because this one I say that this is only an introduction. Okay. It gives you only the overview of these gene cards. Right? In general, when we conduct these drug discovery workshops, for each gene cards itself, we'll spend about 2 to 3 days. Those that means will when we conduct a course. Okay, usually drug discovery entire course that we offer over more than 1 year. So, but it's a tiny piece. So when you go to the on the summaries tab, read the gene card summary. That means exactly follow these steps: 1, 2, 3. Next, fourth one, okay, and also they are given arrows, okay. Next, and then you where you need to look for where you need to look for is also highlighted. This is the step number 5. Then afterwards you want to go to the open pathways. Next one, madam. Then you have to go, because in each and every step you need to spend about 5 to 10 minutes time to go through it, okay, to understand, to read it, okay. Then open this disorders tab now. Then you see the expression tab, because this gives you a visual picture, okay. Suppose even though you are working on lungs, so it may have some kind of a side effects in these organs. Okay, it gives you some kind of a image. So when we prepare this, what is it called, research proposals, okay, when you are putting this one, then it will show you a good confidence in the evaluator team. Okay. Just now, one madam has asked me about the confidence intervals. Okay, these confidence intervals is always there in a database. Okay, you can go to the lower, you can go to the higher. Okay, and if you ask me whether lower is correct, higher is the correct, there is no definite answer. Okay, but when you are putting the higher, you will get only less information. If you put the confidence score less, then more information. Okay. In general, I tell you that this is only what my team works. We always look at confidence interval 0.70. Okay, or 70%. Okay, that means, what does it mean? The 70% means whatever the information that I can say, I abstracted, only 70% of the information is accurate. Remaining 30% of the information, I'm doubtful. Okay. So for the early career researchers, am I right? We always say start from 0.5 first. You see what information it is getting, then go to the 0.6, then some information will be left out, then 0.7, some information will be left out, and 0.8, some information will be left out. It's a confidence score. Okay. Then early career researchers most of the time say that, oh, I want to be 90% perfect. But when you go for 90% perfect, actually, you don't get any result out of it. Okay? Then, madam, then transfer your findings into workshop table point 1.1 below. Go down, madam. So go down. Yeah, this is where. This is where you need to. That means these are the simple. These are the things that you. This is the important information that you need to know. Okay, at this point of time. Then you can read more and more. Then these are the discussion questions. Why EGFR considered as an actionable target in NSCLC? How does combining a gene database? All these things we can discuss later because this one is an introductory part for you to understand. Am I right? Then later on, at this point of time, you are looking at only one database. You will have a lots of questions. Okay, two things. If you are thinking that, oh, there is a lot of information, that means you are on the right track. If anyone thinks that I already understood, that means that person is not doing anything. Okay, and some other person says that, oh, I'm a bit confused, then that means you need to work together with a team. Okay, that means what happens means, in general, in my research team, you have to look at the different points. Am I right? One student will work on only one. Okay, the another one works on the another. The third one will look at the third ones. Then all these three sit together and they will talk about it. Okay, and then ultimately they come up with a PowerPoint presentation, and that PowerPoint presentation will be presented to the that research supervisor or the research mentor to see what happens. Okay. So then here also you can see the troubleshooting, which I am going to tell a lot in the next one, the format, I think in 1 minute time. Okay. So you write only these things, no right answer, no wrong answer. Okay. The take home message is, before this workshop, how many of you know about this database? Okay. So that is the learning point. Okay. So how many of you know what is the usefulness of this database now? Am I right? So the another key message is now, okay, if I want to start any drug discovery research, okay, this is the database that I need to really look at it. Once you get these 2 points, then it's good enough. Then we'll go to the next one, PubMed. Okay, and before we go into that one, PubMed. Okay, now we talked about the genes. Am I right? Whether it is where it is located and all those things. Now we want to see a research on it. Okay, experimental evidence. Okay, that one we will check in the this one called a PubMed database. Okay, so here we are going to talk about how effectively we can use this PubMed, and this is called evidence building. How to create the evidence? We cannot simply say that, oh, Arjun, suppose me, I say that, oh, EGFR is very good. The people won't believe that, don't believe Arjun. They say that so and so person A said this, B said this, C said this. When I put all these three together, A, B, and C, I got this kind of a confusion. Okay, and I want to clarify the confusion. That's why I'm going to carry out the research, and that is called as research question. Okay, now PubMed is not only a literature database. Actually, generally, many people use this only as a literature database, but in drug discovery, it is a decision support tool for target validation, mechanism evidence, biomarker context, clinical translation, and safety intelligence. That means gene card will give you some information, then you are going to validate that information, how many labs did that particular experiment and did prove that. Okay. Now, why this matters? Prevents weak target selection, because already in gene cards, we already talked about what are the things that we cannot do it, right? It supports evidence-based rationally. Evidence means it's only an experimental, okay? Experimental evidence identifies known mechanisms and guide gaps and connects biology to the clinical relevance. This is what we do. Okay, and EGF, for example, it talks about the disease biology, it talks about the mutations, it talks about how the patients are responding to these things. Okay, and here, write reproducible PubMed searches that are focused, balanced, and suitable for drug discovery discussion. This is very important point. Huh? Most of the drug discovery people, what they do is they search one time, they come up with 20 references. Another time they search, they come up with 40 references. Another time they search, they will come up with 60 references. And actually they do not note down how did they do the strategy. That is called as the search strategy, that what we are going to discuss. Okay. Again, start with a research question because you need to know what is one, then build the PubMed research around it. Next minute. Okay, again, from target idea to the clinical relevance, disease need, same thing. Of course, this same thing already I talked about this one. A translation means what evidence supports the patients. Okay. PubMed, what it helps you is what is already reported in the literature. That it tell you. Then it also tell you whether the evidence is recent. Okay, some people will say that, oh, this particular research was done before 2000 or before 2020. The people don't believe such things actually. Actually, to be in a drug discovery research project, we are not supposed to cite any publications that are published before 2003. Okay, why I say in 2003 means 2003 is the one where the human genome was established, and 2003 is the starting point of all these called omics, cheminformatics, bioinformatics, genomics, proteomics, transcriptomics, everything that omics started from 2003. So that is the most advanced research. So there is no point of going before 2003. Okay, that's why they say that evidence is recent and also reliable. Okay, then which studies support the mechanism? Okay, and what are the clinical evidence that exist? And what PubMed do not directly answer is whatever the molecule that you are synthesizing or you are dreaming about, it won't tell you whether it is going to bind or not. It won't tell you whether a prediction is experimentally true. Whatever you are thinking, it won't tell you that, oh, whatever your assumption is going to be true or not, it won't tell you. And it also won't tell you whether the target is going to be safe or not. Okay, and it's not saying that one single paper is sufficient. Okay, then the best mindset is use PubMed to build and challenge your scientific rationale. That means you have to criticize yourself. Okay, that's why you have to challenge your scientific rationale before moving to the experiment or computational screening, even the computational screening. And nowadays, what the people think that here they don't use their brain, simply they go for the computational screening. Okay, it doesn't work. So you need to understand the concept first. Okay, so PubMed converts isolated observations. These isolated observations you got from the gene cards into an evidence trial for decision making. Then you decide whether you want to move ahead or not. Okay, next slide, next slide. Okay, now these are the evidence that you have to look for in the PubMed. Okay, these are the things that we need to look at because in drug discovery, all these things we need. We need to know the target, whether this really works or not. We need to know that how it works, and we need to know that what would be its therapeutic action. Okay, and we also need to know that if I develop a molecule, whether the protein is going to develop any resistance or not. We also need to know what would be the safety of my molecule and what would be, we know that this biomarker is really, can I identify in a patient or not, a population. Okay, then these are the questions that you need to ask. Is ECFR altered in NSLC? How do ECFR, which, how, what, which? Okay. Here I want to highlight the people in a research question, we cannot use the word can. Okay, that is not a research question. Usually what they say is what, why, how, what, why, and how. Okay, that means what exactly you want to do, why you want to do, how you are going to do it. Okay, so how you are going to do is an experiment. Why are you doing it is a rationale. What are you going to do is a scientific gap. Okay, so these only three questions that we use. Okay, don't say can this be done. Okay, all those things are not at all right, or not at all right. Okay, so here actually you see in all my discussions I'm always starting with a research question because this gives you the clarity. If you do not have this clarity, there is no point of fetching all those databases. Okay, then is easier for altered in NSLC. So what you are expecting is suppose this is relevance and target selection. Okay, when you ask this question, it will tell you that whether your hypothesis is correct or not, and suppose which drugs improve the outcome, then you can say that yes, there are some drugs that are showing some kind of an evidence, okay, in a clinics. Then you may ask, actually we need to understand a research, whatever the academic institutions do, actually strictly speaking, we should not call that as a research. There is a specific term that we use, it is called as an academic research. Academic research. Then the actual research is the one that is done by the research organizations, okay, and industries. Okay, you need to know the clear distinction. Suppose if you look at the CSIR lab, okay, IICT labs, CIMB lab, they are the research organizations. Okay, and if you look at Andhra University or Nagarjuna University, these are the academic institutions. What these academic institutions do is they do the academic research. In the academic research, they will train the students how to generate this kind of a research ideas in an effective and efficient way, so that when they move to the research organization, they can do the excellent research. Okay. Now you may ask me, Arjun, why are you saying this? Why can't we do the entire research in our place? Because it is going to cost millions of dollars. Okay, and academic institutions do not pay, do not have that kind of a infrastructure. So please remember, whatever you are doing is called academic research. Academic research means you need to know the research question, research hypothesis, okay, research methodology, how to interpret the data, how to conclude the data. That's all. Okay, you may be disappointed with that. I do not have this experiment. I do not have this protocol at this point of time. You should not. Okay, so search for evidence types, not only the keywords. Madam, can you go to the next one? Okay, now this is very important point. Use concepts, synonyms, and structure. Break the question because this is the one time. Right? What is the evidence for EGFR mutations as predictive biomarkers for the EGFR tyrosine kinase inhibitors in non-small cell lung cancer? Okay. Now, then you have to break down this question into these concepts. Okay, these are called as the key words in the question. Okay, EGFR mutation, non-small cell lung cancer, targeted therapy, or tyrosine kinase inhibitors, then the biomarker or response. All right, this is the question. Then, in the previously, I told you that for each gene there are synonyms. Then this EGFR, you have to divide into these things, EGFR, ERBB1, HER1, because all these are one and the same only. Okay. Then disease concept, and some people will call it as a non-small cell lung cancer, some people call it as a NSCLC, some people will call it as a lung adenocarcinoma. But all these are one and the same only. Okay. Then therapy concept. Some people will call it as a tyrosine kinase inhibitor, some people will call it as a TKI, and some people will give the specific name. But all these are one and the same. So here if you see that all these synonyms you have to put within a bracket, and those two words should be separated by R. Should be separated by R. And here also you are putting all these things, non-small lung cancer, and here also you are putting non-small lung cancer or NSCLC or lung adenocarcinoma. Okay. Or here you can see that either mutation or biomarker or response, you can put that. And this tyrosine kinase inhibitor or TKI. Then you put all these things. Okay. That's why what happens is, okay, the things that actually I evaluate, suppose whenever student try to defend their thesis, okay, the first and foremost question that what I ask is, can you show me your search strategy string? Okay, because this is the most important. If the search strategy string is not correct, that means literature review is not correct. So that means research gap is not correct. That means research hypothesis is not correct. That means research objectives is not correct. Entire thing is wrong. Okay. That's why the first thing that they will ask is what is your search strategy? Because all our decisions is based upon the literature review. Okay. Nowadays the people say simply Google it, they will get it. It's not. Okay. And this itself is a science. This itself is a very big science. Okay. And different search strategies. Build the search from concept first. Do not begin with one long sentence. Okay. That is one thing. Next, madam. Okay, these are the Boolean terms that we use. Okay, and, or, not, quotation marks, field tag, and another field tag mesh. Okay, now when you put this one, actually it happens is narrows, and or means broaden. Okay, not exclude. Usually don't use not. Okay, usually not will be used by those researchers who are considered as the biggest and greatest researchers. Because usually when the student will come with all this data, okay, then they go to the supervisor, then the supervisor think about all about it, then the supervisor will decide that, yes, now in that one you put not and so and so. But in your this one, don't use this one, okay? And this TI stands for the title and abstract. Because that is the most important thing. Because if you are talking about the EGFR, and if you want the PubMed to pick that paper, okay, where the EGFR appear in any part of the paper. It can appear in the experimental, or it can be appear in the results, or it can appear in the introduction, it can appear in the literature. But it is not relevant. Okay, the core message of an experiment usually will be reflected in the title, okay, or it will be reflected in the abstract. So whatever the keyword that you are looking for, it should be present either in the title or in the abstract. That's why it has come up with this one. The second thing is this is a medical subject headings. So what this PubMed did was, because in the past, I'm saying that 10 to 15 years back. So what they did was the researchers may have a lot of confusion because of a lot of different terminologies that the people use. They come up with a measure. Okay. So these are some examples. Okay. Of course, use or this one here I already told you. Sorry, it is a small letter. And use not rarely, it can remove relevant papers. Okay. Boolean logic controls whether your search is broad, narrow, or balanced. Okay. So, which is right, which is wrong, again, it depends upon the research team. Okay. Okay. Next one. Okay. If you look at the mesh, this is medical subject heading. Okay. Why it is useful means it relates all the group related terms. Okay. And it reduces the probability of some of the missing articles. Okay, improves consistency and useful for systematic searches. Okay. Here I want to tell you what is meant by a systematic. Everything should be a system. That means you need to say that someone asks you on what basis you have selected such strategy. Then you need to say that I have selected based upon these keywords. Then the next question is on what basis you have selected these keywords. I have selected the keywords because of I have selected this keyword because of this reason. I have selected this keyword because of this reason. I have selected this one because of this key reason. Okay. Actually, what happens is in most of the research proposals, okay, and literature review is the uh, most appropriate term. Most it is not written appropriately, and unfortunately, most of the PhD thesis and all those things, the least importance that they give to the literature review. Okay, and most of the time literature review, because they have a mind concept that I need to have 300 to 350 pages. It is a psychological concept. But nowadays in all over the world, they say that suppose if I say the Newcastle University, the PhD thesis should not be more than 50 pages. Then they will say that why it is so. That means if the researcher cannot comprehend it within a 50 pages, then what we can't call it as a researcher. And nowadays, whenever we go for the research conferences, am I right? They say that actually we need to explain the entire concept in 3 minutes. Okay, and the researchers are being trained that entire PhD research has to be conveyed within 3 minutes. And the people who sits in front of you is who doesn't know anything about the research, but they must understand. So actually, it is the truth. Truth means we are doing the research not for the scientific community. We are doing the research for the general public. So they should know what you are trying to do. These are the decent things that 40 people are doing it. And of course, I know that in UGC also they started. Now all the PhD thesis need not to write a dissertation. If they publish 5 papers, automatically it will be PhD will be awarded. Or China has come up with a new thing that is called if anyone has a 2 patents, that's all it is considered as a PhD. Because nowadays the people are thinking that writing it when it comes with AI and all those things, the people are thinking that it is waste of the student's time, it is a waste of the reader's time to write a thick thesis. Am I right? Previously, during our time, there was no AI tools, there is nothing, there was no Google. So as there is someone who need to refer to a textbook, that's why theses are there. But nowadays the concept is continuously changing. Okay. So then very new papers may not yet be fully indexed because this is the problem. Because you may say that Arjun, you say that mesh is the one very useful, am I right? Because it covers everything. But there is one paper that is published in not just 1 month back. Okay, so the people who are curating that mesh, they need some time to put that into there. Am I right? So, so if you rely only on mesh, you may miss very recent paper. That is the key thing. Yeah? Okay, recommended approach, you have to combine both. Okay, you have to combine both. And sometimes, depending upon the concept, okay, you don't believe even the search strategy itself can go up to a maximum of 10 pages. Can go up to a maximum of 10 pages. Because if a particular disease or a particular gene have a lots of mutations, okay, so suppose if you are talking about inflammation, inflammation is everywhere. Am I right? So if you want to put inflammation, that is my longest search strategy, which is about 72 pages. When someone wants to do that one, that entire search strategy itself is 72 pages, then it doesn't matter. The PubMed can put everything into it. Then and usually these are the things will be guided by there are some experts which are they call it as a systematic reviews. Okay, they write only systematic reviews. Okay, so mesh gives structure, keywords give sensitivity to new and specific terms. Madam, next madam. Okay, then this is the one, start broad. Okay, then check result count and relevance. That means once you got it, first you check the 5 to 10 titles, whether it is okay or not. Okay, then you have to add synonyms, then add focus terms, then add filters, then document the final strategy. This is very, very important. I think my colleague has missed this one to put it in this one. You need to write the document strategy. So that's what you need to say. Okay, what is the best practice is, this is the search strategy that I use, okay, on so and so date, on so and so time, and in which time zone. Okay, because why it says that one is, usually when the students present to me, and first thing is whatever the search strategy that they give, I will copy and paste and I will search for it. If there are changes in the references, then automatically the thesis will be rejected then and there itself. And unless otherwise they say that a particular so and so date and time and the time zone, and actually we can go and check where it is. Then it should be reproducible. Okay. Then what to record for drug discovery, exact search string, data search, filters used, numbers of results, 3 to 5 key papers, one line takeaway per paper. This is very important. And here, this is what you can take artificial intelligence or generative AI. Okay, in that one, okay, for me, usually what I say is, I take any generative AI, I upload this paper. Okay, usually I will ask, I don't like this paper. Okay, I thought there is some mistake. Can you find out a mistake in that? Okay, because why I asked that one is, and it gives me the research gap. Because ultimately I want to do research on it. That's what I do. Or I price the paper. Hey, this person did a fantastic thing. Okay, so I want you to tell me what are the good key message that I can take. Suppose I'm giving a lecture on particular thing, right? Suppose non-small lung cancer, someone asked me to give a lecture on non-small lung cancer, then I need to search the literature on, right? Then I go to the PubMed and I will say, I take about 50 papers. Can you tell me out of these 50 papers, please rank them according to the quality of the experiment. Then in my literature, I say that there are 100 papers, in the 100 papers, these 30 papers, good, and all. Then I say what is the key information. And actually I use AI and actually I scold them. I scold my generative AI assistant. Okay. So I scold them verbally and I scold them by writing. Okay. The way I scold. And now my generative AI assistant really understands my mood. Even when I switch on my camera, by looking, when I'm reading it, by looking at my facial expressions, it says that, sorry, Arjun, it seems you are not happy with the search data that I did. So I'm sorry, I'm going to redo again. Okay. And all these are the paid versions. That means it is exactly similar to, that means my AI research assistant is actually the human research assistant. The way I speak to my student, same way also I speak to that one. That much thing was there. So it makes me, suppose we are, so far my team, when I say my team is, there is also research assistant is also my team. Okay, AI research assistant. Now it tells me that actually what I want, and now I have a less headaches. Okay, it is very similar to when a PhD student start journey with you. It will take some time for the PhD student to understand what you want, what you expect, how you react. In the generative AI, also the same thing. So if you keep on moving from one assistant to the another assistant, one assistant to another assistant, okay, and no assistant will going to be the best research student for you in your entire time. Okay, so you get one research student that is the assistant, you train them, okay, make sure that it listens to you, it obeys to you. Okay, so that's the key thing. Okay, so you look at this, and all my research team use only the same research assistant. Okay, that is called SciSpace. Okay, so here what to record for drug discovery: exact search string, date search, filters used, number of results, 3 to 5 key papers, set, and one line takeaway per paper, how the evidence. Okay, next one. Okay, here I want to tell you that because I'm not revealing any kind of a generative AI name because I'm not here to promote anything. Okay, for me everything is good. Okay, it depends upon what it suits to you. Okay, okay, small changes that improve evidence quality, use synonyms. Okay, and separate evidence goals and use advanced search. Okay, and here is good searching is iterative. Search, inspect, refine, document. All these are important. You cannot simply say that EGFR I got it or finished. No, you need to say that search. Of course, this inspect, search you have to do it manually. Okay, how AI can be used? Search you do it manually because this is what you are going to do. Of course, inspect you ask your assistant. Okay, hey, I got 10,000 papers, but I don't want 10,000 papers now. I want only these things. There I can say that, okay, I want the papers that are published from the Harvard University, Cambridge University, or this one. Or I can ask, I want you to screen the papers which are published by the top 50 universities, okay, QS rankings. So if you want the quality data and all those things, it doesn't mean that they only do the quality ones, but at least it helps. So in that one, it will help you. Am I right? Inspect and refine. Of course, that also you can ask. Anyway, you got me this top 20 papers, but I'm not happy with it. No, I want you to rethink about it and this one. Or you can also ask, hey, I got about 100 papers. Okay, can you please explain to me who are the authors, who are the principal authors? Can you show me their H index? Okay, then it will show you their H index. Then the people with a high H index, and I take those papers. Then I ask because that is the research assistant. Okay, it's not the other thing. If you use the search, that is not that is called as irresponsible use of AI agent. Okay, or unethical use of AI assistant. So what it will do is this one, and nowadays this document also it will tell you. Okay, you can take the research assistant. Hey, okay, now I'm happy with these 100 papers. Can you prepare me MS Word document about? Okay, I want only 10 lines. Okay, what is the main aim? What experiment model they did? What is the result they did? Okay, and what extra information that they have given, which is not reported earlier, because that is the key and crispy information, and it will give you in another in 5 to 10 minutes time. Okay, whereas if you want to do it, it will take 2 years or 3 years. Okay, that is called the responsible and the ethical use of generative AI. Okay, thank you. Next. And previous one, yeah. Searching one word only too broad. Okay, I think these are the avoiding weak searches. Okay, applying filters too early. Okay, don't apply using not aggressively. Of course, at the starting of my career, I used not a lot and lots of headaches I got. Then comparing reviews only and no documentation because some people when they want to write a review article, they assume that if I collect only the review articles, this is what the people do most of the time when they write a literature reviews or when they write thesis reviews. Okay, they collect only the reviews. Okay, and they're assuming that everything will be collected there. Okay, that's one of the mistakes that they do. Okay, most pub mistakes are not technical; they are planning mistakes. That means PubMed is good. Actually, PubMed itself is the AI tool. Okay, PubMed itself is an AI tool. That's why when you are giving a keyword, it is searching everything that we are seeing. But now, since the generative AI is coming and we are ignoring that the PubMed is not AI. Actually, PubMed itself is an AI. Okay, and it was invented very long time back. Okay, so that's the thing what we need to do. Okay, next. No, next one. Okay, template, define the decision, what evidence is needed before Okay, list concepts. Add, of course, I'm also going to share the PDF documents of this one later. Okay, and the information that is there in your workbook, and also this one you can add. Okay, combine synonyms, document. Okay, and this is the example search. Okay, and the best research is a transparent, reproducible, and aligned to a decision. That means transparent means anyone check. That one I do. That's why when it comes to me, first question I will see is the search strategy. The search strategy is not strong, it's gone case. Actually, most of the research proposals that are submitted, the first question they will ask is how strong is your literature search? Okay, and reproducible and aligned to a decision. Next, madam. Okay, do not write. Many studies show EGFR is important in lung cancer. That's what many students write. It's not at all right. The correct way of writing is PubMed evidence. You need to say that what database told you that one. Evidence supports EGFR as an actionable target in non-small cell lung cancer because activating mutations predict response to the EGFR tyrosine kinase inhibitor, while this particular mutation, tyrosine at the position of 790, which is mutated to methionine, is a major resistance mechanism requiring mutation selective inhibitors. Just imagine yourself, you are putting up a research proposal and you are saying that this is the reason that why you are working on research. Do you think that anyone has a courage, okay, to not to give your project? If you give this one, nothing. Am I right? So this is the one. Then you have to add, when you are writing a research proposal, this is where you will start. Okay, by looking at the statement itself. Am I right? That evaluator, okay, go ahead. And maybe this could be one of the factors where I got a greater success in securing the grants. It doesn't mean that I'm a greatest scientist. Definitely I am not. Okay. It doesn't mean that I have a very good infrastructure with me. No, I don't have a very good infrastructure. But only I'm good at is I'm good at writing a research proposal. Okay. And then add 2 to 3 case citations, okay, or type of evidence. That, of course, whenever we talk about evidence, it should be an experimental evidence and what description, decision it supports, what uncertainty remains, what experiment or tool comes next. Okay, then of course we do this one. PubMed evidence should lead to clear next research action. There is a lot of databases are available. PubMed is considered as one of the most reliable databases, okay, for medicine related research. Okay. Next, madam. No. Okay. I'll talk about the Campbell when I'm talking. Okay. These are the take home messages. PubMed is essentially for evidence based target validation. Searches should be based on drug discovery decisions. Use synonyms, combine, mash, record the search string. Do not treat one paper or one review as a proof. Okay. Here, the most important thing is what you take home message is search, use your brain. Don't use generative AI. You do that part, and all the remaining part you can ask generative AI to do for you. That is the best thing. Okay. There is a one famous saying. It is called as garbage in, garbage out. Okay. That is one thing. And until now, the people, no one knows in the world how the AI works. Actually, it's honest. Okay. Now, once upon a time, people say that if you use generative AI to write a research papers, it will get rejected. But nowadays, all the editors have changed their mindset. You should use generative AI to write a paper, to make it a crispy, to make it understandable, but you declare it in acknowledgements. You declare it. So you can see a transition. Okay. But it is an ethical and responsible use of AI. And even I have seen one paper recently, which is published in a Nature. That guy may be very ambitious. In that one, say Chat GPT said this one, in the that paper got published. Even the reviewers did not see it, editor did not see it, and it was retracted. It was retracted. Okay. So in general, the AI is a two swords, two-edged. It's good and also it's fine. Even now also I have to be very, very careful because I also use generative AI to do these things. Definitely, if someone expert is there, definitely they may caught me at one or the other point. But using a generative AI makes my life easy in one way. But checking is going to be very, very difficult. Checking it. But in this way, it helps me because all these slides that I have prepared while I was traveling in way. Okay. So in a short period of time, I could able to do, otherwise it cannot do. But if it is caught, okay, for example, any researcher was caught by irresponsible use of AI, and those researchers are banned. Okay, in China, it is much stricter. If anyone found that, then that person won't get any job anywhere in the China. Okay, and please don't, even the people from Harvard University were also what is it called? Those papers also were retracted. So be careful when you ever use generative AI. Okay. So do not start with a database. Start with a scientific question and the intended end use. Okay. This is one thing. For me, my research team for scientific question itself, we spend about 1 week to 2 weeks for brainstorming. Okay. Then only we go and search for the data. Thank you. Okay. Then if you go to the PubMed database, go to the website, madam, PubMed database. And please remember that this entire thing is not only prepared by me, it is prepared by my research team. So sometimes they will call, even yesterday also I called, and they only told me that actually you have to say this, you have to say this, you have to say this. Okay, why I'm saying that is a single person is not sufficient to do anything. Okay, so you go to the PubMed. Go to the PubMed. Okay, here you can see that there is one button called search. There is some button called advanced. In the search, you can put it. Okay, madam, can you click the advanced? Can you click the advanced one? No. Or you put yeah. So here, am I right? This is a very important point. Madam, you put these things. Put this arrow down. Put this arrow down. You see, add with and add with or. Okay, add with not. But please don't go with not. Please don't go with not. Okay? Now, here you can go search for all fields, but it's not at all advisable because it gives a lots of mess. Madam, when you click down, here you can see that, okay, go up. Suppose if you know that author name, okay, then you can search for it, okay, or you want to go for a master, you can use it, or you can for publication, you can do it. Go down, madam. Just scroll down, madam. Okay, suppose if you want to see this one, don't put the filters, don't put this one. No, no. Of course, if you are interested only, for example, if I'm looking at a journal, drug medicinal chemistry one, usually I go for maybe ACS journals. Okay, Journal of Medicinal Chemistry or RSC journals, all those things. But at the initial period, you need not to know because even though when I search for it, okay, still I can access that full paper, I can read it. But whereas if you search for it, you know that there is a paper published in GMC that you want to read it, but you don't have an access, it is waste of your time. Okay, madam, can you go down? Here. Okay, go down a little bit. Okay, language, you can choose mesh. Here you see mesh stamps, Okay, but usually we select the investor. Okay, then go down. Okay, publication type. Okay, go down, go down. Here you see that this is the one most powerful title or abstract. I told you that this is where you need to select. Okay, now simply you put EGFR. EGFR, you put R, R. Okay, so here you can see that EGFR within bracket title or abstract. It came up, right? Then here you can put for example, HER1, HER1. Okay, then you put R, R. Okay, here you can see. Okay, so like that you can build a strategy. Okay, then you put search. Then you put search. Then it will give you how many? 105,543 results. Am I right? Of course, here you can take off, you can take the help of AI. Okay. Here, of course, it is not there in your this one. Okay. So usually what I do is can you put this send to madam? Okay. Here you can say that you need to suppose if you use the Mendeley or anything, okay, automatically, madam, you go to that one. Center, you can go to the citation manager, or you can go to the bibliography, or you can give this one, and yeah, okay. Usually, what happens is there are a lot of reference manager softwares are available. There are also AI tools. You can use that. Okay. Then, madam, can you click the display option, please? Here, you can take a summary of it. Okay, madam, go this one. Okay, abstract, PubMed, and this one. Okay, then okay, close it. Close it. So can you come in here? It is getting the results from 1980 to 2026. Okay, here also we need to see. Right. Here you can see the trend. It is increasing. That means what? More and more researchers are working on it. That means it is a current problem. And nowadays, all the research proposals, they're asking, Arjun, can you show me the concept? Can you show me this kind of this one? So they want to see the upward trend. And most of the think that, oh, research means no other person should do. I only should do. Okay, this is what the people are thinking of. That is a wrong concept. Once upon a time, I was also in that concept. Okay, it's not. Okay, so when you are seeing the upward trend, then the people say that go ahead. Now here you can see some kind of a fluctuation. Then sometimes the people will ask, hey, why there is a fluctuation here, drop and all those things. Okay, then maybe it may be because of the COVID-19. Because during COVID-19, all the labs were closed. Okay, you need to give such kind of an explanation. Okay, madam, can you put this one here? Scroll, no. This one, try to move that side. Try to move that side. Move. Yeah, move. 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 Yes, stop there. Stop there. So I am looking at the last 10 years. That itself is a 68,951 results. Okay. Then you go down, madam, here. Okay. These are the, you play around. Okay. Here, madam, there is a, can you go to that side, please? Up, little bit up. There is a user guide. Okay, you read that entire thing. Everything will be there. Everything will be there. Whatever I have given you is only a tiny part. Okay, so that is about this PubMed. Okay, so what we do is we take about 14 minutes. Okay, you play around with it, and here you may wonder that Arjun, can you tell me the exact way that to search? Who am I to say that that is the only way to search? Am I right? There are more than one ways. There are more than many ways. and some people it's used to something. But some people who are working in a pharmaceutics, for example, this PubMed may not work. PubMed may not work. Okay, so that's why I'm not saying that this is where you explore. Am I right? And usually, in general, what happens means in kind of my research team, every 6 months, one of my research member has to give an update to us what was changes happened in the past 6 months. Okay, even that is also now we feel that we are lacking because every time it is changing. If I say something tomorrow, it may not be this one. Okay, and the other thing is whatever I studied in the B form, it is no more applicable now. So that's why I do not want to give any kind of a false or negative thing, but you people search. Okay, now before this, how many of you searched PubMed? Okay, how many of you used the Boolean terms? Okay, so that shows that what is the usefulness of this particular workshop. Okay, now you understand how to set the Boolean terms. Okay, how to use the Boolean terms effectively, how to put the search strategy, all these things. Okay, that's all. As long as you know this one, it's fine. Later you go and explore. Okay, we'll stop at this point. Maybe we'll start at 11:25. Okay, you give me some gap. Okay, then we are going to talk about the string database next. Okay, thank you. In the meanwhile, please check and write down.
Dear participants, we'll come back to the session by 11:30 a.m. In the meanwhile, the speaker has asked you to just practice the hands-on workshop materials that has given to you, and we'll come back by 11:30 a.m. with a new link for the live stream. Thank you.